Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. This opportunity, I would like to present our research, the title Antagonistic Activity of Volatile Organic Compound of Innovative Bacteria from Small Breakthrough Teres ensiformis against soil-borne fungal pathogen by Dr. Abdul, Munif, and I, Prayogo Probo Asmoro, from Department of Plant Protection, Faculty of Agriculture, IPB University. Okay, we know that plant disease control is plant and pest disease are a limiting factor in crop production and that cause decrease in the quality of quantity of agricultural product. And then the one group of pathogen that are mostly reported because disease is from fungi, and some fungi are soil-borne pathogens, and then they are relatively difficult to control. And some example from important soil-borne pathogen are Solani or Rhizoctonia solani, Sclerotiomyces E, and Fusarium sp. And there are the three fungi also have a wide host range that make it even more difficult to control. And plant disease control is not directed toward a higher level control that more environmentally friendly and have a minimum negative impact. One of the most developed plant control agent is innovative bacteria, and Asmoro and Munif 2019 success in isolating innovative bacteria from Teres ensiformis, and 4 of which isolate were able to control the pathogen of Rhizoctonia solani, which cause rice-seed-blight disease. And innovative bacteria have a various mechanism in controlling pathogen. One which this mechanism is producing a volatile organic compound, and then the study about this mechanism have not been many reports regarding the production of a volatile organic compound by innovative bacteria. So the research objective was to evaluate the antagonistic activity of UCS produced by innovative bacteria from the spot breakthrough against 3 soil-borne pathogenic fungi, Rhizoctonia solani, S-E, and Fusarium sp. And this research consists 4 stages. There are isolation and preparation of the innovative bacteria, and then isolation and preparation of soil-borne fungal pathogen, and then evaluating of inhibition activity of innovative bacteria against pathogenic fungi, and then GCMS analysis, and last is data analysis. And then in this research, the isolates used in this research, there are APE 15, APE 22, APE 33, and APE 35. They isolated from the roots of Teres ensiformis by Asmoro and Munif 2019. And then the result in this picture we can launch. We know that the 4 innovative bacteria could produce volatile organic compound, and then the inhibition was indicated by the colony diameter of 3 types of innovative bacteria that is smaller than the consular colony. This result so that they indicated could produce secondary metabolite from volatile organic compound that have antagonistic activity against 3 pathogenic fungi. The colony code inhibition of the 3 types of soil-borne pathogen began to appear from day 1 after inoculation until day 5. And then in general, the 4 isolate or poor treatment had a high inhibitory ability to the growth of colonies of three types of soil-borne pathogen through the producing volatile organic compound that has antagonistic activity. The high inhibition for endocritic bacteria occur in rhizobacterium and then followed by Sclerotumor E, and in contrast, the low suppression occur in Fusarium sp. Isolate APE15 was the best isolate in suppressing the growth of Arsolani and Fusarium sp, while the isolate with the highest suppressing ability against Sclerotumor E was the APE33. And then the GCMS analysis was conducted to determine the active component contained in the endocritic bacteria, especially from isolate APE15, because this isolate have a relatively better ability to suppress of three types of soil-borne pathogen compared than the other isolates. The GCMS results are shown in figure 5, and then the chromatogram in the viticulture isolate APE15 showed 25 detected peaks. The detected peaks were then analyzed with a mass chromatospectrophotometer, and each peak from mass spectrum was matched with chemical abstract surface data and referred to the compound in this table. Some of the dominant compounds detected were octanol, 19.4%, and then followed by cyclohexane propanol acid, 12.34%, and silane, 8.53%. Some research report that octanol and the other compounds like acetra and alpha-terpinol could significantly inhibit the other pathogenic fungi like Clostridium citae aurantii, and the antifungal activity of octanol can disrupt the integrity and cause lack of cell component. Octanol in several concentrations was reported to inhibit the growth of Penicillium digitatum spore in citrus fruit. So the conclusion is the study provides information that the endophytobacterium isolate from the root of Teres ensiformis can act a purifying agent by suppressing the three types of soil-borne pathogen by producing volatile organic compound. And then octanol compound from APE15 isolate is thought to be FSSAT by antagonistic activity against the three soil-borne pathogenic fungi. That's all. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.